So today my topic is about myocarditis. So as you know, heart is not a very beloved organ by immunologists, but recently myocarditis become a kind of issue. So while I was almost finishing this work, I had a chance to meet Dr. Drew Weissman, who was a Nobel Prize winner of 2023 about his invention of mRNA vaccination. So he mentioned about a lot of stories, and he said there are a lot of conspiracy theories about the mRNA vaccination recently. So he was suffered from that kind of story, and he said he can admit only two types of side effects can be scientifically reasonable. First one is, of course, anaphylaxis. It's all vaccines side effect. And second one is mentioned as myocarditis. So I thought my work is valuable, and I was very happy about the original inventor of this vaccine is also recognizing the problem about the myocarditis. But the first problem when we study myocarditis is, when we face is, how can we define the myocarditis? Of course, we can define by tissue or imaging, but it's usually very vague, and it is very difficult to get this kind of a tissue from real-world patient because of the safety. So let me introduce very briefly about the background of myocarditis. So let me name the conventional myocarditis before COVID-19 era. I would like to name it as a conventional myocarditis. So myocarditis can present with a range of clinical manifestation, including chest pain and other chest symptoms, but usually it's okay, usually known as it's okay. But someone also have very higher mortality if someone is complicated with heart failure or arrhythmia. And the etiology of conventional myocarditis is usually known as a virus, but usually in the real world, we cannot diagnose the real etiology because viral PCR positivity rate is relatively lower in real world. And about the immunopathology of the conventional myocarditis, monocyte is the critical role. And also there's an interesting phenomenon called the emergency myelopoiesis, which is the egress of immature monocyte from the bone marrow in certain circumstances in mouse model. And during this phenomenon, CCL to CCL2 axis was shown to be very important in some myocardium-related mouse model. But it's all about the animal model. There's no human evidence about that. And vaccine-associated myocarditis these days is very hard, and different pathogenesis compared to traditional conventional myocarditis is expected. One of the potential mechanisms for the occurrence of vaccine-associated myocarditis is extended free spike proteinemia in the blood. So as you can see in this clinical paper, after vaccination, myocarditis patients have prolonged spike proteinemia in peripheral blood. So purpose of this study is to compare the commonalities and differences in immunophenotypes of tissue-confirmed myocarditis between COM and vaccine-associated myocarditis. So I focused mainly on this vaccine-associated myocarditis first, but there's two different types of controls used. First one is conventional myocarditis, kind of disease control. And second one is normal vaccinated control with the same age and gender profile compared to this VAM-related patient. And I gather PBMC with some longitudinal way, and also I have chance to access to the explanted heart tissue who underwent heart transplantation due to this myocarditis event. So we did single-cell transcriptome and ATAC, TCR, BCR analysis, and spatial omics technology. So this is brief characteristics of cohort. And important thing is all of these myocarditis cases were confirmed by tissue confirm and also with MRI confirmation. So I tried to confirm, enroll the real myocarditis cases based on clinical information. And when we look into the blood cells of myocarditis patient, one thing very interesting was we could find a decrease of monocyte and increase of CD4 T cell. And when you look into the transcriptome changes in qualitative way, myeloid cells underwent the most dramatic changes based on transcriptome level. So that's why we focused on myeloid cells at first. So when we look into the myeloid cells, monocytes of the myocarditis patients, we found very interesting population that we call this CCL2 positive classical monocyte. So these monocytes are extremely deviated only in myocarditis patient, and almost it was very minimal in non-myocarditis vaccinated control. As you can see, the expression of CCL2 is very high, and also CX3, CL1, and some degree of IL-1 receptors, and they don't express the usual monocyte marker called the CXCR4, and so on. So I think this kind of marker can be a very important blood-based immunologic diagnosis of myocarditis, because it can be a substitute for the myocardial biopsy or some dangerous or expensive process. And we validated this finding using flow cytometry as here. And interestingly, this myocarditis-associated CCL2 classical monocyte proportion is positively associated with WBC count, neutrophil count, CKMB, the cardiac enzyme, and LDH count. So that means degree of inflammation and degree of tissue damage is positively related to the proportion of CCL2 classical monocyte. And so I decided to look more into about the tissue macrophage profile of this patient. We have a chance to look into heart tissue, and we gathered all the macrophage and monocyte cells from the myocarditis patient. And interestingly, the most of the heart infiltrating myeloid cells also have CCL2 positive classical monocyte features, as you can see here. And when we look into more detail about this CCL2 expressing classical monocyte, interestingly, they have a very immature feature and like a stem cell feature of the recently bone marrow migrated features of monocyte. So as you can see here, when we look into the pseudotime analysis of this classical monocyte, the CCL2 monocyte is in the very early phase. And when we look into the cell cycles, it's dominantly in G1, has higher degree of S phase and G2M phase. So relatively, CCL2 monocyte is immature and freshly egressed from the bone marrow, and they are related to the recently known phenomenon called emergent myelopoiesis. So I'd like to deep dive to the mechanism why this kind of cells and what is the origin of this cell. So when we did ATAC sequencing using this myocarditis patient sample. So based on the ATAC profile, interestingly, we could find some novel cluster called the CCL2 positive myeloid progenitor. So we already found the CCL2 positive classical monocyte in this myocarditis patient. But as you can see here, there is a separate population called the myeloid progenitor. So the reason I named this cluster as progenitor is they have a lot of expression, open chromatin area on GATA 1, 2, 3, and 6 area, which is very important transcription factor to regulate the stem cell property of myeloid precursor cells. And when you look into the details of this CMP population and chromatin openness of the CCL2 region, we found that it is very close to CCL2 positive classical monocyte, but they also have some enhancer region openness. So that means that CCMP is very close to CCL2-CM, but they also have some increased area of the enhancer and so on. So when you look into these areas close to the CCL2 proximal regulatory regions have very wide opened in this CMP, and as you can see in the right side. So this is the characteristics of the myocarditis common, and I described about this focused on CCL2 classical monocyte. And then I'm trying to see the difference of VAM and COM. So interestingly, this vaccine-associated myocarditis people has monocyte with a higher frequency of CCL2 intermediate classical monocyte, and CCL2 high monocyte is higher in conventional monocyte. And the expression level of CCL2 is generally higher when compared to vaccine-associated myocarditis. So we compared these two different types of myocarditis monocyte using transcriptome level. So as you can see here, normal monocyte is here, and the vaccine-associated myocarditis dominantly monocyte is here, but the conventional myocarditis monocyte is dominantly reside in here. So this PC1 can define the characteristics of myocarditis-related monocyte cells. So PC1 is, as you can see, dominant PC1 negative population has a lot of interferon-related signatures were increased. And when you look into the PC2, which divide vaccine and conventional myocarditis, as you can see here, PC2 positive, the vaccine-associated myocarditis has additional overexpression of TNF-related NFκB pathways and so on. But for the conventional myocarditis, usual interferon-related pathways were only activated in this population. So when we look into this in more detail, similar findings were observed. So I can define that myocarditis in general is related to interferon response, but vaccine-associated have some additive features called the iron beta and TNF-alpha responsive features, and not about COM. And this is a similar story. But one thing very interesting in clinical finding is in vaccine-associated myocarditis, there is a steep dip of absolute monocyte count in the all acute phase of this myocarditis. So there must be some reason that monocytes are dying because of some molecular reason or some inflammatory cause. So my hypothesis was it is related to inflammasome activation, and it can be related to pyroptosis. So I decided to some in vitro experimentation to see what can cause the activation of pyroptosis or inflammasome activation. So as you can see here, LNP and spike protein can induce some activation of inflammasome, but they have also additive effect. And as you can see, spike protein alone can activate inflammasome-related pathway, including NLRP3. So, and it is also related to pyroptosis of those monocyte cells. So I think this pyroptosis and inflammasome-related pathway is also additionally activated in the vaccine-associated myocarditis. And quickly look into the T cell part. Inflammasome-associated cytokine milieu governs the Th17 pathology and clonality in vaccine-associated myocarditis. Interestingly, in vaccine-associated myocarditis, there is a strong expression of pathological Th17 genes, including OX40, GITR, and GPR65, which is well known from the animal models of Th17 researchers. And these are also the supporting evidence that Th17 activation is more prominent in vaccine-associated myocarditis. And finally, I look into more about the T cell. So so I had chance to have a longitudinal cohort in some part of this patient. So I had hypothesis that tracking T cells with identical TCR in real-time longitudinal samples, we can discover some interval changes of identical groups of T cells. The reason I applied this hypothesis is this finding, this negative finding. When I look into the acute and differences between acute and late phase of the myocarditis, there is definitely no difference between this acute and late phase myocarditis transcriptomes. So I was very sad, but I had that kind of hypothesis there. If I compare same TCR clone in our body, it will be a very fair comparison, because that same group of T cells still exists during the observation interval in our body. And my hypothesis was right. When we look into the only shared clones of the T cell in CD8 T cell or CD4 T cell, these shared T cell clones have definitely higher frequency, higher score of TCR activation module or bystander activation module. And when we look into this VAM and COM related changes in transcriptome using this volcano plot, only for the TCR same clones, I found that there's a very interesting finding there. So VAM has generally consistently high TNF iron signature, as I mentioned, but they also have a hype of interferon signature in acute phase, as you can see here. But COM has consistently have high interferon signature, as I mentioned, but they also have hypo TNF iron beta signature in acute phase. So this iron TNF NFκB related signature plus interferon signature is kind of pathologic hypoinflammation at acute phase. So I was very interested in this kind of two different SIT pathways intervening because these are well known as a pathologic hypoinflammation. When I was a PhD student of Richard Shin, so we have a chance to publish this kind of COVID-19 related hypoinflammation phenomenon based on these two intervening phenomenon, and also about this, also proved this in the ferret animal model before. So not just for the infectious disease, but also for the hypoinflammatory response to vaccine can be also explained by this kind of interaction. So these are the final summary. So we discovered the myocarditis specific CCR2 monocyte in patient blood and tissue. And I think it's interferon induced and related to emergent myelopoiesis. So immature monocyte is egressed from the bone marrow. Etiology-driven divergent pathomechanism can be a temporal convergence. So in active phase, they both are activated, but interferonopathy is general in COM. Inflammasomopathy is general in VAM. Suggestions for clinical activity is vaccination interval. Epidemiologic studies showed a higher myocarditis risk in second dosage and lower risk in delayed subgroup. So this delayed spike-proteinemia can contribute to the risk of myocarditis. So if someone already experienced a weak degree of myocarditis, he or she can use mRNA vaccination with a prolonged interval. So that kind of hypothesis is also possible, I think. Final one is suggestions for using immunosuppression agents, including corticosteroid as anti-interferon and iron blechoid in severe myocarditis cases. So thanks for the, thank you. Yeah.
So great work.
Thank you.
Obviously not everyone gets myocarditis. So have you looked at what identifies the individuals that may go on to get the myocarditis? So is it some genetic risk factor in those, or is it this issue of administration where young men, with big muscles, there's a risk of an intravenous injection of the mRNA? So what do you think about why is it only a subset of the at-risk people actually get myocarditis?
I received the same question from 3 audiences in this room already. So the short answer is I don't know. So, but I tried these things for this study. First, we had a whole genome sequencing data for this cohort, and we could not find any kind of meaningful germline mutation from this myocarditis cohort. As far as I know, this is the largest cohort with biospecimen for the myocarditis, but we couldn't find. Second, I tried to find some kind of a somatic mutation or other epigenetic or post-germline data, but we couldn't find anything. We tried to find deep sequencing for the whole genome DNA of the monocyte of this patient, but we couldn't find any kind of differences from other populations, and failed to find epigenetic regulation features, specific features for this patient. The only thing I found was the deviation of a monocyte and the specific population called the CCR2. And the previous step for emerging this CCR2 monocyte is the key is the interferon response, and also combined with NFκB related things. And I think these NFκB related things is related to those inflammasome activation originated from spike protein or other due to the prolonged exposure to the spike protein. But I don't know reason why they have a prolonged spike proteinemia, especially in healthy young male.
Yeah.
I don't know.
Very nice talk.
Thank you.
I assume you had both mild and severe patients because some of your tissue samples are coming from extracted heart tissue. So did you try to compare mild versus severe patients to identify some differences between them?
Oh, very good question. But we decided to only focus on severe myocarditis patient because, as you can see here, everyone had heart biopsy result. That means they had some necessity to confirm the diagnostics. So that means they all had relatively severe cases. And in relatively milder cases, it is almost impossible to get heart tissue and confirmation. And I believe some myocarditis cohort with milder symptoms actually have no myocarditis in some cases. So for clear comparison, we didn't include myocarditis with mild clinical manifestation.
Thank you.